Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Let me talk about why we care about HIV. It's one of the deadliest pandemics in history. Treatments are effective in the first world, but in the third world, they're very complicated and not really effective. So if we want to do something for the disease for now most of the people who have it, we have to come up with something like a vaccine. But vaccines are difficult because HIV mutates, not just like, I don't know, seasonal flu mutates, but it actually mutates in the body of the patient to make itself more deadly over its course in a single patient. So a hypothesis in our group, and in general, is that there are a few spots of vulnerability. This is the DNA of a map of the DNA of HIV. A few spots of vulnerability in the DNA, and they depend on the immunity types of the patient. If our immunity system just kind of goes on its own to try to learn how to come up with some attacks on the DNA, it'll pick what's basically random spots. And those aren't likely to be the spots where the HIV is actually vulnerable. If we have a whole protein vaccine that will train up our immune system, it will still just be training up our immune system at spots within what it covers, and those also aren't likely to hit the parts of vulnerability. And that would be like the Merck vaccine. What we want is a vaccine that specifically targets the areas where we think there's vulnerabilities to train up the person's immune system so that they can be ready to fight the vaccine if they get it, or the virus if they get it. And we have some evidence that this approach can work. This is data from the kind of famous Merck vaccine trial. On the bottom is the number of places where the vaccine helped a patient attack the virus. And here is the amount of virus in the patient's blood. You can see there's no real relationship here, nothing visible. But if we take a look at the predicted vulnerability points, and these are points that our group predicted were vulnerable before the Merck results were available, we see that in cases where we predicted there were no vulnerability points covered by the Merck vaccine, the HIV, the virus load was very high, and in the places where there was one vulnerability predicted by our results, the virus load was lower. So this gives us hope that we're on the right track. So I'll continue by talking about our group. A big part of our group is scaling. So I'll talk about how we kind of just practical experience in scaling across processors, computers, users, and very briefly, users and computers. And then finally, just one slide of the web tools that we've open sourced, or the applications we've either made available as web tools or open source, or in most cases both. So we're a small group. We're applying computer science to other branches of science in health, energy, water management, and so on. We tend to partner with experts who have the wet labs and clinics, Los Alamos, Harvard Medical School. That's nice. We don't, at least I personally am glad we don't have wet labs there in Redmond. The part of the group that's working on these kind of HIV and disease-related projects include David Heckerman, Jennifer Lisgarden, Jonathan Carlson, Bob Davidson. Bob and David are here, and Nebosha. And the goal of the group is to produce first and foremost scientific progress in the form of treatments, and is measured perhaps by journal articles. We produce software tools that we can share. And we also serve a useful function by giving feedback to other parts of Microsoft on what scientific users need from their software. So the part of the group that I'm in started out as part of the machine learning group. And a lot of what we do is create new statistical algorithms that exploit something that previous statistical algorithms didn't have. And that's just tremendous amounts of computation. In the past, there were often kind of theoretical ideas in statistics that if hypothetically you could say, look at lots and lots of permutations, you could set some value, but it wasn't practical to do it kind of in that theoretical way. Well, now that theoretical approach has become practical in many cases. So we're exploiting both increasing amounts of data and increasing amounts of computation. And of course, like any statistical software, the purpose is always to get the most signal that we can from the data. And the second thing, of course, is to supply these tools to all interested users. So let's look at one problem we had and kind of the tools we used to, the approach we used to solve it. So we have a program not yet shared called Eigenstrat covariance. And kind of what it sounds like it does, if you have a lot of patients, say 1,000 patients, for every pair of patients, you want to come up with a single real number that expresses the similarity of those two patients as measured by how much they have in common in their DNA. A run, typically, if you just kind of run it the normal way, would take 3 hours. And that's kind of boring. You have to start working on a whole other project. You lose track. Your productivity goes down. We'd really like, if we have an 8-processor machine, to get the result 6 times faster, something like that. And what we found very useful is parallel extensions. And basically, well, we'll see in a second what that is. So it's a new standard component of .NET. It'll be coming out in Visual Studio 2010, but a version of it's now available as a technology preview. Its whole point is to make the easy things easy. I think there's been a lot of work in parallel programming to make the hard things possible. And what we're discovering is there's so much easy stuff that it's important to make that simple to do. We use it primarily at kind of the first and last steps of the kind of programs we write. A lot of times there's either preparation work, data cleaning, or some kind of summarization step that is not practical to run on lots and lots of machines, that kind of has to run on one machine, perhaps because it needs quick access to all the data. And it's nice if you can be using all your, say, 8 processors to do that. And let me just give a simple example. Suppose we just have a 2D array and we want to find the mean of every row. Kind of regular C# programming is here. We just loop over the rows I, and then for each column in J, we total things up, and then we find the mean for each row. So with the parallel extension library, here's the change we have to make. We've basically only changed one line. Now we just say Parallel.For. The only thing you have to do to be careful here is we have to be sure we put our results in a pre-allocated array of doubles. And these pre-allocated arrays are thread safe, so we can just be putting in the results as they come in any order, and we'll have the results there. And the whole parallel extensions library has many, many components, but we basically find 3 to be to do 99% of what we want. Parallel.For, which this is an example of. There's Parallel.For each, which works exactly the way you'd expect it to work, given that you've just seen Parallel.For. And one very interesting other feature, which is more different and has some additional power, parallel link. So with parallel link, you can create a for each kind of statement. You can do groupings, where tests like we saw in the previous talk. The nice thing about it is you don't have to have a pre-allocated place for your output. It'll do the work in parallel, but it'll give you the results in a single sequence, kind of one at a time as they're finished. So let's look at another problem. PhyloD is a program for finding associations between viral DNA and some other factor like a user's immunity type. It's a typical run can take a CPU year, and that's too long even on 8 processors. So for that, we run it on the HPC, and we have a library that makes things a little more convenient. And I'll talk about that briefly. So one version of PhyloD is just this command line program. Say PhyloD, you give it the DNA of the HIV. You give it this phylogenetic tree, this tree of family relations between the DNA sequences. You tell it the HLA of the patients that you're giving it the data for, and you give it an output file. So this is just something you could run on a regular machine right at the command line. What's nice is that it's delightfully parallel. Every line of output in the final report is independent of every other line. So to make this run on the HPC, we just have to change it in kind of a MapReduce way with just, as a programmer, just supporting a few additional command line arguments. First, task count. So you have to add code to tell it how to break the work up into 1,000 parts. And then tasks, you have to tell it what parts this particular run of it should be working on. So we add the code to break, so we can break the work into 1,000 parts. And in this case, we want it to work on part 22. And we change the output file to have some kind of pattern thing there. So when we get these 1,000 output files, they won't all have the same name. And finally, we code something when we type -cleanup, and all that has to do is merge the different output files. And that might just be merging the output lines literally, or sometimes there's a more reduced kind of thing where there's some statistical analysis done, but relatively quick compared to the production of the output. The simplest case, the HPC already comes with a relatively nice graphical user interface. And if you have a job set up like this, you can just tell it to run the 1,000 jobs to divide the work and the 1 job to combine the results. And we also have a library. And if you link into our library, you can just add one command in the minimum. -cluster, where you tell it the cluster and where the data is on that cluster if they're the same place. And with just that additional command, your job that did run on your own desktop now can run on the cluster, eventually get the results, and have your, in this case, single output file waiting for you on the file share. So we had another program. This program only takes like 15 minutes to run. It's called Create Epitome. It merges amino acid sequences together to create a short vaccine that gives you as much coverage as you decide is worthwhile. So our problem was we wanted to make this a web app, and 15 minutes per user doesn't seem that bad, but if a dozen users start using it all at the same time, and you're running it just as a web app, then the web server is going to start complaining, and your web hoster is going to tell you can't run your app anymore here. So in this case, we wrote it. We created a version of it in Silverlight. Here's a snapshot. This works nicely. From the user's point of view, it's a web app. It runs on Macs or PCs under IE or Firefox. You still have a GUI that's pretty much as nice as a desktop GUI. Especially good from a programming point of view is adding the Silverlight, making the Silverlight version, and since the original code was in C#, was basically almost no extra code. And we had to write code for the GUI, but that was useful for the desktop also. And best of all, it now does the actual 15 minutes of computation on the user's machine instead of on our web server. And finally, just one thing was alluded to this morning. You also have the scenario where you have perhaps hundreds of users and you want to run on hundreds of processors. What do you do then? We now have, I'm told, available a version of PhyloD running on Azure that addresses this as an alternative to you sending your data to David or me or Jonathan and having us run it for you. So just list a catalog of the things that we have available now. There are more things. We have programs and applications coming out all the time. And a concluding statement that there are new statistical methods powered by increasing computation for fighting HIV and other diseases. The thing that powers this ability to use increasing computation is the ability that the libraries like Parallel Extensions, HPC, Silverlight, and Azure give us to kind of efficiently code up these new programs. And for more info, there's a link to all the bio apps, and you can go to my web page, which is a little easier to remember, and you can see the Codeplex and the web pages.
Thanks, Carl.